Hello everyone, today I'll be discussing the topic tryptophan operon. To begin with, the tryptophan operon is found in prokaryotes like the E. coli bacteria, and this refers to a group of genes that encode for the biosynthetic enzymes that are required for the synthesis of tryptophan. The tryptophan operon is expressed when tryptophan levels are low and it's repressed when the tryptophan levels are high. This is regulated by the TRP repressor. When it is bound to tryptophan, the TRP repressor blocks the expression of the operon. So tryptophan biosynthesis is also regulated by attenuation, which refers to a mechanism that is based on the coupling of transcription and translation. Thus, when the tryptophan is naturally available in the environment, E. coli can take it up and use it to build proteins. But when it is not available, E. coli also has the ability to synthesize tryptophan using the enzymes that are encoded by the 5 genes that comprise the tryptophan operon. The TRP operon includes 5 genes that encode the enzymes needed for tryptophan biosynthesis along with the promoter to which the RNA polymerase binds to and an operator which is the binding site for the repressor protein. Adjacent to that are the 2 regions called the leader and the attenuator. The genes of the tryptophan operon are transcribed as a single mRNA, and this stretch of DNA is recognized by a regulatory protein which is known as the TRP repressor. When the repressor binds to the DNA of the operator, it keeps the operon from being transcribed by physically getting in the way of the RNA polymerase, which is the transcription enzyme. There is the presence of another gene known as TRPR, which encodes a repressor located at a distance away from this gene cluster. So this is the structure of the TRP operon, and which is the operon of E. coli and some other microorganism, which contains the 5 main structural genes which are designated TRPE to TRPA. So these 5 genes encode for 5 polypeptides that bear the 7 functional domains necessary for tryptophan formation. These genetic segments correspond to 2 pairs of functional domains are fused and they yield the bifunctional polypeptides TRPG to TRPD and TRPC and TRPF. Now the mechanism of the TRP operon. The TRP operon is responsible for the production of the amino acid tryptophan, whose synthesis occurs in 5 steps and each require a particular enzyme. And these enzymes are translated from a single polycystonic mRNA. Adjacent to the enzyme encoding the sequences is the presence of the promoter and operator and the 2 regions called the leader and the attenuator. The transcription is turned off when a high concentration of tryptophan is present and it's turned on when tryptophan is absent. The regulatory signal is the concentration of tryptophan itself. The TRP operon has 2 levels of regulation, an on-off mechanism and a modulation system. The protein product of the TRP gene, which is the TRP aporepressor, it cannot bind to the operator. However, if the tryptophan synthesis has to occur, the aporepressor and the tryptophan molecule together join to form an active repressor complex that binds to the operator. When the external supply of tryptophan is depleted, the operator becomes exposed and the transcription begins. So this type of on and off mechanism activation of the aporepressor, which is as a product of the biosynthetic pathway, has also been observed in other similar pathways. When the tryptophan operon is repressed, this is usually the case unless the concentration of tryptophan in the medium is very high. The optimal concentration of tryptophan is maintained by the modulation system in which the enzyme concentration varies with the concentration of tryptophan. And this modulation is affected by the premature termination of the transcription before the first structural gene is reached. And the second factor is the regulation of the frequency of premature termination by the concentration of tryptophan. And located between the 5' end of the TRP mRNA molecule and the start codon of the TRP gene is a 162 base segment called the leader sequence. And within the leader is a sequence of bases which have regulatory activity. So after the initiation of the mRNA synthesis, most of the mRNA molecules are terminated in this region, and this yields a short RNA molecule that consists only of 40 nucleotides. And this terminates before the structural genes of the operon. The two tryptophan codons make the translation of the leader polypeptide sequence sensitive to the concentration of the charged tRNA TRP. If the tryptophan is limiting, there will be insufficient charged tRNA TRP and the translation will pause at the tryptophan codons. So the biosynthesis of TRP depends on two characteristics of gene regulation in the bacteria. First one is the transcription and the translation are coupled. The second one is the base pair formation in mRNA should be eliminated in any segment of the mRNA that is in contact with the ribosome. Next is the regulation by the TRP repressor. The TRP repressor does not always bind to the DNA. Instead, it binds and blocks the transcription only when the tryptophan is present. When tryptophan is present, it attaches to the repressor molecules and changes their shape so they become active. A small molecule like tryptophan which switches a repressor into its active state is called a co-repressor. On the other hand, when there is little tryptophan in the cells, the TRP repressor is inactive and it does not attach to the DNA or block transcription, and this allows the tryptophan operon to be transcribed by the RNA polymerase. So the TRP repressor acts as both a sensor and a switch. It senses whether tryptophan is already present at high levels and it switches on the operon to the off position. So this prevents any unnecessary biosynthesis of tryptophan. So attenuation is similar to the TRP repressor mechanism and it reduces the expression of the TRP operon when the levels of tryptophan are high. However, rather than blocking the initiation of the transcription, attenuation prevents the completion of transcription. So when the levels of tryptophan are high, attenuation causes the RNA polymerase to stop prematurely when it's transcribing the TRP operon. So only a short mRNA is made and that it does not encode any tryptophan biosynthesis enzymes. So attenuation works through a mechanism that depends on coupling, that is the translation of an mRNA that is still in the process of being transcribed. So there is a section that lies between the operator and the first gene of the operon that's called the leader. The leader encodes a short polypeptide and this also contains the attenuator sequence. The attenuator does not encode for the polypeptide, but when transcribed into mRNA, it has self-complementary sections and it can form various hairpin structures. Once the RNA polymerase binds and starts transcribing the operon, a ribosome can attach to the still forming transcript and begin translating the leader region. The polypeptide encoded by the leader is short and just 141 amino acids long and it includes 2 tryptophan residues. And this is important because if there is plenty of tryptophan, the ribosomes won't have to wait long for a tryptophan carrying tRNA and can rapidly finish the leader polypeptide. If there is little tryptophan, the ribosome will stall at the TRP codons and it will be slow to finish the translation of the leader sequence. So the leader sequence is followed by the attenuator region. which can stick to itself to form different hairpin structures, and one structure includes transcription termination signal while the other does not end termination. If the ribosome translates slowly, it will pause, and its pausing causes the formation of the anti-terminator. And this hairpin prevents the formation of the terminator and allows transcription to continue. So when tryptophan is abundant, the ribosome moves quickly along the leader sequence and the terminator hairpin forms and the transcription of the tryptophan operon ends. When tryptophan is scarce, the ribosome moves slowly along the leader and the transcription of the tryptophan operon continues. So in other words, the logic of attenuation is same as that of the regulation of the TRP repressor. In both cases, the high levels of tryptophan in the cell shut down the expression of the operon. So this makes sense because high levels of tryptophan means that the cell does not have to make more amount of tryptophan.